It's now my pleasure to introduce our final speaker, next generation leader Denise Cai. Denise received her PhD at UCSD, where she characterized how REM sleep enhanced creative problem solving by integrating recently learned information with prior experience. As a postdoctoral fellow at UCLA, she discovered that memories encoded close in time are linked by sharing an overlapping neural ensemble, such that recalling one memory triggers another temporally related memory through the overlapping ensemble. Denise is now an assistant professor in the neuroscience department at the Icahn School of Medicine at Mount Sinai. Her lab investigates the temporal dynamics of how memories are formed, integrated, and separated. In addition, her team studies how aging contributes to cognitive decline. Please welcome Denise Cai.
Thank you. Thank you, Justin, for that lovely introduction. Can you guys hear me? We're good? And I think you just recapitulated my whole talk, so we can just go to happy hour, no problem. Thank you so much for having me here. It's really a pleasure and a privilege to see the impressive science that you guys are doing and the way that you guys are doing it. It's really inspiring, and I'm trying to think of how we can incorporate some of these philosophies back in my lab. So today, I'm going to take the time to talk a little bit about my postdoctoral work at UCLA, how we link memories across time, and then also a little bit about the Miniscope project, which was our venture into developing neuroscience tools on an open source platform. So one of the amazing things about our brains is the ability to have so many experiences across a lifetime, integrate it into our brains, and somehow efficiently and effectively recall experiences correctly. So for example, one of my favorite memories that I like to recall is my wedding day. And I might think back to that day when my dad walked me down the aisle, and he promised me he would not cry, but he was just so moved by the person I was about to marry that he couldn't hold himself together. And thinking about that experience might then trigger a subsequent memory where here Tristan and I, for the first time as husband and a wife, get to toast her family and friends, forget about wedding planning, we're ready to live happily ever after. And thinking about that memory might then trigger a subsequent memory when our guests fully took advantage of the open bar at our wedding reception. Somehow, my brain is able to temporally relate these events together and separate them from other experiences in time, such as here, my husband and I are toasting to a different occasion at Napa Valley. So the question I'm going to pose today is, how is it that our brain is able to take these different experiences and temporally relate them together and separate them from experiences that happen distinct in time? So what do we know about how the brain encodes multiple memories? Most of what we know comes from a very rich literature in associative learning. And typically in this paradigm, there's a CS and US, such as a tone and a shock, and they co-occur, boom. And they converge in areas like the amygdala, which then leads to behavioral responses, such as freezing, in this case with Pavlovian fear conditioning. But in the example I just gave you, these experiences happen across time, across hours, within a day. And so there isn't a lot of information about, in the literature of how we encode across these time scales. So while we don't know a lot about how we encode lots of memories, we have learned a lot about how we process single memories. Oh, I'm supposed to give you the take-home message. It's been a long day, so Justin already told you the take-home message, so he stole it from me. So what I'm going to tell you today is that the way we think we link memories close in time is that they share an overlapping neural ensemble, such that when I think about this experience that reactivates an ensemble, but because of the overlap, then it triggers this temporally related event. Okay, so what do we know about how single memories are processed? So canonically, we think that learning is encoded in a sparse population of neurons, and such that at later recall, when you re-experience that event again, many of the same neurons are reactivated. And then when you experience a different event, a different population or an independent population of neurons are activated, such as in hippocampus, and this is how we tell memories are similar versus if they're very different. So when I was in Alcino Silva's lab, one of the questions that we had was, why is it that this particular neuron got to encode the memory compared to its neighboring neuron has seemingly very similar inputs? Are there rules that bias memories to certain populations of cells? Now, Sheena Jocelyn and others have shown that the allocation is not random and in fact has very specific rules. So for example, her lab showed that if she injected CREB, which is a transcription factor that's important for learning and memory, into a very sparse population in the amygdala, then when she trained the animals on tone conditioning, the cells that express CREB were the ones that were more likely to recruit the memory into those neurons. And when Yu Zhou was in Alcina Silva's lab, what she did was patch onto these CREB-positive neurons and found that those neurons that had CREB also had increased cellular excitability. They're more ready to fire. So that led to the very simple hypothesis that CREB, this transcription factor very important for learning and memory, led to increase in excitability, which led to allocation of the memory. And so when I had joined the lab at the time, that was kind of where we were. And a number of us thought, well, can we just bypass CREB, and just go directly to see if increases in excitability can then allocate the memory. So in studies led by Thomas Rogerson and Balaji Jayaprakash in the lab, we took advantage of opsins, and thank you so much, Andre, for explaining set function opsin. So what we did, similar to the experiment that Sheena did, was inject a sparse population of neurons in amygdala with a virus that contained both step function opsin, which will just depolarize the cells a little bit, and then a redshifted channel rhodopsin in which we can cause the cells to fire. So what we did was, prior to presenting the tone and the shock, we shined blue light to depolarize the cells with step function opsin. And then later, we wanted to see if we can optogenetically recall those cells. If we reactivate those cells that were previously active, will we recall the memory? And so in a novel environment, we don't play the tone. All we do is artificially reactivate those cells again. And indeed, what we did see was behavioral representation of memory, such as freezing. So this and a number of other studies, and Sheena's lab also showed very similarly using chemogenetics to activate those cells. They got very similar results. So together, that suggested that excitability alone was enough to allocate the memories. So that was very exciting at the time. But then we thought, you and I, we encode memories all the time. We're not injecting viruses. We're not shining light into our brain. And just because you can artificially cause the memories to go to those cells doesn't actually mean this happens endogenously. And so we began thinking, well, we know that excitability endogenously fluctuates across time. And we began to think, could this be a mechanism then to link memories that are encoded close in time and separate memories across time? So we know that one way that excitability is modulated is through learning. And John Disterhoff and others have shown that after learning, there are transient increases in excitability that comes up on the order of hours, and across days goes back to basal levels. So this gave us a really nice, simple framework to test our hypothesis about how excitability might allocate memories and link memories together. And so first, we had to confirm that indeed it was the cells that encoded the memory that had increases in excitability. So we turned to Mark Mayfield's TET-TAG mouse, which has an activity-dependent promoter that's fused to a GFP. And so we're able to, after learning, just a context learning, we're able to tag those active ensembles with GFP, and we patched onto those cells to show indeed, in those cells specifically, there was an increase in excitability at 5 hours. Okay, so great. Now this gave us a framework to test our hypothesis. So the prediction then is that if a second experience is encoded when excitability is high, then that context should be encoded in a very overlapping population of neurons, such that when excitability comes down to basal levels, another experience would be encoded in an independent population of neurons. So the predictions we tested was that memories within this window of increased excitability indeed are co-allocated or share an overlap of neurons. And as a psychologist, I really care about the functional consequences. It's just how I was trained. And I want to ask, well, why would you ever want that to happen? Why would you want to have this overlapping population of neurons? And we hypothesized that it was through this overlap that you're able to co-recall temporally related events. And then the third thing that we wanted to test was a hard test of our hypothesis, that we predicted it's because of increased excitability, and I'm going to be looking at CA1. Then in disorders with deficiency of excitability in CA1, we should see an aberration in linking. So that's what we tested. So to look at the neural ensemble activity across these long periods of time, we developed a miniature microscope system at UCLA through the open source platform. In the last few minutes, I'll tell you a little bit more about those developments and kind of the genesis of how we did that. But this was really inspired by the technology developed in Mark Schnitzer's lab with the miniature microscope system. And this was a very collaborative development, so not quite as big team as you guys are doing here, but it was a collaboration between 3 labs. Okay, so this is the very first version of our miniature microscope system imaging dorsal CA1 of hippocampus. On the top right, what you see is the raw calcium transients, and this is the DFF, so it's, we aligned it, and this is just showing the change of fluorescence over the mean fluorescence. So using this technology then, we can have access to thousands of neurons in dorsal hippocampus and see how they encode these experiences across time. So what we did was, while we had the animals explore 3 very different contexts across 7 days, separated by 7 days and 5 hours, and we imaged the neural activity of dorsal CA1. So here is just a representative animal. This is the mean raw image. And so what you can see is that we can see the same blood vessels across these time periods, and we use this to align our frames. And here I just kind of pseudo rainbowified just so that you can see the different cells. And while on average we saw about 600 cells active during the exploration of 10 minutes in these contexts, there were different cells coming in and out to represent each of these 3 contexts. So based upon the hypothesis I just told you, what we then would expect is that there would be a higher overlap of these 2 contexts, because they're separated by 5 hours when excitability is high, compared to the overlap of these 2 contexts separated by many days. So that's what we looked at next. And indeed, what we found was that just by having 2 contexts pushed closer in time, there was a higher overlap in the neural ensemble compared to if they were separate in time. And so this was some of the first evidence suggesting that just time is playing an important role in how memories are allocated. So that's cool, but so what? What does that mean? And how is this functionally related? So we wanted to, oh, sorry, this is just to remind me to tell you that we think that this mechanism is actually generalized across brain regions. Sheena Jostlin's lab published a beautiful paper in Science showing that in the amygdala, with 2 different conditioning stimulus, they also show a higher overlap when the 2 experiences are separated by 6 hours compared to across days. Okay, so next we want to ask, what is the functional consequence of having this overlapping neurorepresentation? And the hypothesis was that then thinking about this experience through that overlap is going to trigger thinking about this experience. So how do you test this in mice? So similar to the experiment I just told you about, what we did was we had animals explore 3 different contexts, so each row here is just a different group of animals. And now what we do to probe its behavior is we bring them back into the blue box to give them a shock. And then we test them in one of 3 contexts. Okay, so what do we think is going on in the minds of the mice? So when they go back to the blue context, we expect that they remember being in that blue context, they were shocked, and they think, oh, shiznits, and they freeze. And this is their natural response to being afraid. Now, when they go, and that's what we see here, some moderate freezing levels. Now, when they go back to the green box, we expect that they recall the green box, reactivating the neural ensemble. They weren't shocked, so they're exploring, hanging out, and so they freeze less, which is what we see. Now, the interesting question is, what happens when they go back to the red box? They were never shocked in the red box, but there's this overlap of the red box and the blue box in which they were shocked. So we hypothesized that when they go back to the red box, they recall the red box, but thinking that red box activates memory of the blue box, and they think, oh, shiznits, and they freeze. And that's exactly what we saw. Despite never being shocked in the red box, they show fear in that red box. Now, some of you might look at this and you say, well, Denise, maybe your mice are just not very smart. Red box, blue box, it's all the same box. They can't tell the two apart. And so to try to address this question, we asked, well, if they really think the red box and the blue box are the same, then we should be able to interchangeably use one to extinguish the fear of the other. And so to do that experiment, what we did was very similar to the last experiment. We had the red and blue paired 5 hours apart. We now know there's this neural overlap between the two. And then we bring them back to give them immediate shock so that we have a behavior readout. We test them in either the shocked box, the 5-hour non-shocked box, or a brand new novel box. So here we replicate the results where we show that indeed they do show fear in the linked box, but this is not a generalized fear. They don't freeze to everything as they, you can see there's much less freezing in this novel box. So this is a very specific transfer of fear. Okay, now we have some fun. We bring them back to either the shocked box, the linked box, or the novel box that's no longer novel over and over and over and over to try to extinguish the fear. Then we give them an extinction test, and what we should expect to see is that going back to the blue box, there should be a decreased freezing, because that's just regular contextual extinction. When they go back to this blue box, because they've never been extinguished in the fearful box, it's just this novel, we should see the same amount of freezing. And then the question is, does going to the red box, that shares an overlapping neural ensemble, extinguish the fear in the blue box? So what we find is, so when they go back to the blue box here, which is noted here, we see there's a decreased level of freezing, normal contextual extinction. By seeing this novel box over and over and over does not extinguish the fear. And interestingly, going into this red box does not extinguish fear to the same levels. So this is some evidence to suggest that despite that they are temporally linked, they are still distinct from each other. Okay, so we show that, yes, there is a behavioral linking. And the last thing we want to test is what happens when there are deficits in excitability in CA1. So we and others have shown that as mice age, there are deficits in excitability in CA1 of hippocampus. And specifically in middle-aged mice, it's not just that there's overall deficits. So if you look at naive mice, before learning, there's no difference in resting memory potential or input resistance. It's only after learning there are deficits in learning-induced increases in excitability. So we thought this would be a really great model for us to test our hypothesis. So we used middle-aged mice. And one of the reasons we used middle-aged mice was we wanted to look at the deficits in memory linking before there's general cognitive deficits. And because we're asking the mice to learn two contexts, we wanted to make sure there was no deficits in learning each individual context. So here, this is just a control experiment showing that indeed in young and older mice, so young mice are 3 to 6 months, older mice are 13 to 16 months, there's no difference in their ability to learn single contexts. Also, we want to make sure that there's no deficits in the ability to reactivate reliably the neural ensemble at recall, and here we show that there's no difference. So we confirm that, at least in these middle-aged mice, they can learn single memories just fine. So now we replicate the experiments and we ask, what happens with the allocation or the overlap between memories encoded close in time? So we replicated in young adults, showing yes, indeed, there's an increase in overlap at 5 hours compared to 7 days. However, in older adults, we found that there's no difference in the level of co-allocation, and there's no difference in the overall number of cells representing the context as well. Okay, so now if there's not this overlap in a representation, that would then lead us to predict that they will also not behaviorally link to experiences across 5 hours. So we replicated the experiment I described before, where we have two experiences separated by 5 hours. We know in young animals, they share an overlapping neural representation. And then we give them a shock, and then we test them 1 of 3 contexts. We replicated in the young mice, indeed, that there is this linking, this very specific transfer to this 5-hour context that does not generalize to a novel environment. However, in the older mice, what we find was despite them being able to learn the single event just fine, they did not transfer that fear to this context that they saw 5 hours before. what was kind of interesting was if you actually look at the amount of freezing in the novel context, the older mice seem to generalize their fear more non-specifically to novel context compared to young mice, which is something we would have predicted. And I have to speculate here that maybe the reason that older mice will generalize more non-specifically is because to compensate for the lack of this temporal linking, the specific linking temporally. But that's just a speculation, I don't know. Okay, so unfortunately, as we age, our hippocampus have deficits in excitability, but this is a message of hope, so we thought, well, if it's really excitability, deficits in excitability, then what if we just artificially increase excitability? Can we rescue it? So we turned to the DREAD system, and here we're using the activating version of DREAD, where, and here's just like an example. We infected about only 15% of cells in dorsal CA1 around the injection site, which equates to only about maybe 1% of all the cells in dorsal CA1. And so this is a very sparse population of cells are infected. And here's just a check that indeed if we inject CNO, this inert ligand, it activates these cells to fire, and this is just shown through immediate early gene expression. And so we know it works in our hands. Okay, so what do? We want to activate a sparse population of neurons prior to learning this red context. And the idea is that if those cells are active, then it's going to bias the red memory, or the memory of the red context into those cells. And then 5 hours later, we inject CNO again to reactivate that same population of neurons to bind those two experiences together. And then in a control group, we give them injection of saline first, and then we give them injection of CNO second. And then we do the same experiment where now we bring them back to the blue context and shock them. And we test them in the never shocked context. And what we find is that by artificially increasing activation in the same population of neurons prior to both events, we're able to increase that behavioral transfer of memory. And that this increase in freezing was not just due to generalized increases in anxiety from CNO injections, as here when we test an elevated plus maze, which is typically used to measure anxiety, we see no difference between saline and CNO. So these results suggest that we can artificially increase excitability and rescue these deficits. And this may have therapeutic implications, as here we're only activating 1% of neurons, and we're able to rescue the behavioral phenotype. And we know that spatial memory is encoded by many parts of the brain, and so maybe just by manipulating hippocampus, we can manipulate downstream targets as well. Okay, so in summary, what I've showed you so far is that memories within this window of increased excitability show an overlapping neural population, and this overlapping neural population allows for the recall of one memory to trigger temporally related events. And in aged mice, where there's deficits in excitability in CA1, we can rescue these temporal linking deficits by artificially increasing excitability. And so in my lab now, we're taking this very simple hypothesis and seeing how this extends to if we have really traumatic events. Do we extend the temporal window of memory? Do we cause overlinking? How do we separate these events? What about for positive memories? So far, these studies, including my studies and others have been published, have been primarily using fear conditioning as a model, but does this extend to cocaine-related memories, social interactions? And so we're starting to explore those other avenues as well. And in my lab, we're continuing to study how these excitability alterations emerges. As I mentioned, here we're talking about deficits in CA1, but the paradox is that in CA3, there's increase in excitability as we age. So how do those two brain regions that project directly to each other interact? Okay, so we think that this is a kind of a fundamental property for how our brain integrates information across time. We also have evidence that this happens in retrosplenial cortex and other brain regions. And so I think it's really interesting to study across these different memory models. For the last few minutes, I'm going to tell you a little bit about our Miniscope project. This is a really fun project, and we were really inspired by the kind of open science that the Allen does, and we try to model some of our ideas from you guys. As I said, this started as a collaboration between Pema Gulshani, Alcino Silva, and Baljeet Coxlab. And how it started, how many stories start with a lot of liquid courage from lots of champagne. And I still remember being in Payman Goswami's lab. We're celebrating a paper, and I was like, Payman, we have some preliminary data about how we think memories link across time, and Alcino and I would really love to be able to track, memories across time. And we're talking about using 2-photon, but I really want to look at naturalistic behavior. And with a little bit of liquid courage, he said, let's just make our own and share with the world. And we're like, oh, that's bold, but we just kind of, went with it. So as I said before, this project was really inspired by Mark Schnitzer and colleagues' invention. They published this beautiful Nature Methods paper in 2011. Then Kunal and some of the other postdocs took that technology and formed a company, Inscopics. And we just thought this was such a transformative technology that we also wanted to develop our own version. We couldn't afford the Inscopics version. I think it's about 2 of them for $150,000. So we decided, instead of spending $150,000 on 2, we spent on some postdoc salaries, and then we would develop our own, in which now we can make them for about $1,000 each or less. So we think about this Miniscope project as at least kind of a 2-part project. So we've developed a system with the head-mounted scope. We have data acquisition hardware, and this all integrates on any kind of computer, and it also can be integrated with the behavioral camera. But we also have spent a lot of time developing this resource, which is our miniscope.org. It is on a wiki backbone. And so we have all of our design files, tutorials. And so for users, you can just download everything as is, use it as is. We've had many people do that. But we also have a lot of developers who take the source code or the 3D design files and innovate on top of that. And we always encourage that kind of innovation. We ask that people bring it back and update the miniscope.org website with whatever innovation that they've added. So this is just kind of a map of the users from our wiki, where in the world they're logging on from. So we launched this wiki last January. Since then, we've had 2,300 registered users, which has really created this really wonderful community where we kind of all help each other through discussion board. We have over 300 labs that are building miniscopes around the world, and we also are doing workshops, hands-on workshops, 2-day workshops where people come and build miniscopes. We do in vivo demonstrations, we do surgery lectures, we go through data analysis. We also have done pre-SFM workshops, which it's a crash course, 3-hour lecture-based. And so now over 750 researchers have attended these 12 workshops across the 2 years. And we're continuing that effort both at UCLA and now at Mount Sinai. So I've been mostly focused on CA1. We've also spent a lot of energy trying to perfect the surgery for many other brain regions, other cell types, different kinds of virus combinations. And so these are some of the areas that we've been imaging over the last 2 years. One of the cool things about the current Miniscope that's available through the wiki is that we've changed the cabling system. So for those of you who have used some of the commercial system, they use a LVDS cabling system, which is many, many cables that have to be braided together. And so here we use a coaxial cable system, which is used in the automobile industry, right? So when you're backing up your car, that's the cable that tells you you're about to hit that car behind you. So this system has to be extremely reliable. We do the Denise test, which is I shake it vigorously like a mouse would, and I can't break it. And what's really nice is that the coaxial cable is compatible with commutators, which allows for much more robust behaviors. So more recently, we've developed a newer version. So we call it the no strings attached miniscope. So this is the wire-free miniscope. This is just our first prototype of our wire-free miniscope of an animal performing a social task. So we know that animals prefer to explore other animals. So they prefer to explore a social cup versus an empty cup. And in this small pilot, behavioral pilot, we just wanted to see, can we get better, more robust naturalistic behaviors? And so in this experiment here, we had animals that didn't wear any scope. They either had a wire-free scope or a wired scope. And what we show is that there's much more robust exploration of the social cup compared to the empty cup when they are not tethered. And so another fun project that we've been working on is looking at how the hippocampus codes large environments. So here is a 25-foot long track. It's so large, we have to have 2 cameras, one on each side, that we combine the video. And here's some example traces from that experiment. And so we take the same animals, we run them on a baby track, a medium track, a daddy track, and we see how their spatial codes evolve across those different environments. So hopefully, we're hoping to submit this soon and that this will be available to the community as well. So my favorite slide, why I love my job and I love coming to work every day, I've had the great privilege of postdocing with Alcino Silva, who's an exceptional mentor, and also Payman Ghoshani was also one of my mentors. And all of the Miniscope developments is really engineered by Daniel Aharoni, who is a physicist. He actually started studying dark matter, and we brought him into the light of neuroscience. Tristan Schumann was a postdoc in Payman Ghoshani's lab. He was not only the handsome groom in my wedding pictures, but also a co-developer of the Miniscope and has his own lab at Mount Sinai with me now. I also want to thank Justin Shobe, who helped with many of the behavioral experiments. And I want to thank my lab, and this is our lab here at Mount Sinai. They're continuing these efforts on the East Coast, extending our family to the East Coast. And thank you so much for your attention.
Thank you, Denise. We have a few minutes for questions.
Hey, there is a difference between temporally linked memories and causally linked ones.
Yes.
It is very interesting that everything you presented were both, in which you had the capacity to move between the red box to the blue box, and then the shock was in there, and never the other way around. And that is question one. The second question comes in regarding your last experiment, in which you have the capacity to activate, to increase the excitability for a tiny subset of neurons.
Yeah.
And if, theoretically, if you can do that, you no longer need temporal proximity for that. Right. You could theoretically do that a week apart, and you no longer need to have both temporal proximity and the same neuronal ensemble being activated.
Yes.
Have you looked at those?
Yeah, great question. So the first question I thought about a lot. You know, why would you want to link memories across time? And one obvious reason, as you mentioned, is maybe because if one predicts the other. And so in experiments I didn't have time to show, we looked at how if you increase negative valence, so instead of 2 neutral experiences, if you increase the valence, how does that modulate the temporal linking window, and is it asymmetric? And what we found in those studies is that if you have a very negative experience, you are actually going to, or in the mice's case, they will tend to link those experiences back in time more than forward in time. And at first, I was very puzzled because there's no biological mechanism that links it back in time, except that evolutionarily speaking, it's much more relevant because the past predicts the future. And so these are the kinds of experiments that we're doing now. And what we're finding is that something happens between encoding and the retrieval of these experiences. And my hypothesis is during sleep, there's some remapping and causing these safe and aversive experience to get bound together if they're causally linked. So great question. So the second question is if they are separate in time, can you just link them by biasing those neurons? So we're doing those experiments now, and hopefully I can give you an update next year. Okay.
You talked about an ensemble coding in memory. What's the role dynamics there? I mean, you talked about kind of these populations that fire every time. Are they firing an identical pattern, or
Yeah.
how much variability is there?
So that's a great question. And the question was, when I talk about these ensembles of neurons that are encoding the memory, what is their temporal sequence? Do they fire in similar patterns? Kind of what is the definition of maybe ensemble, excuse me. So in this case, they don't fire in the same sequence, and probably because this is very exploratory behavior. Mice are not using the same trajectory each time, and so we are seeing that they are play cells in this context, but because they're not going through the same path, we don't see the same sequence of cells fire. However, if we take these mice, put them on a linear track, and we look at the same neurons, we do show that there are spatial receptive fields that are reliable in those neurons. But do they always fire when they come back? No. And so I think what we've showed and what Mark Schnitzer has shown is that when a cell in hippocampus, for example, is encoding a space or a place, it is reliably firing to that place, but it doesn't always fire every time the mouse encounters that environment. And that's how, one way that we are able to tell time through these ensembles.
So I have a bit of a sociology question.
Sure.
So you have this really interesting situation with the Miniscope where you have all these labs all creating data. I'm wondering what kind of dynamics have emerged in the community for handling standards for analyzing the data and analyzing the derivatives of the data and how people educate each other and things like that.
So that's a really great question that I know that the Allen Institute is the leader in maybe answering this question, but I can tell you from our experience what we've tried to do in a very small scale way. So when we published our paper, we had a lot of people that are like, oh, cool, we want to use Miniscopes. It's really easy to see flashing cells. And people send me data all the time, be like, hey, can you just analyze this real quick for me? And as you guys are all well educated, these kinds of analysis are not real quick. And so what we did was I uploaded all the data. So we wanted to just share our raw data so that people have an avenue to practice analyzing or developing analysis scripts for these kinds of data. We didn't have a very elegant way to do it, so I just uploaded everything onto my personal Google Drive because I have an unlimited drive, and then I put that link on miniscope.org website. Recently, I got some emails. They're like, hey, Denise, your folder is empty because someone accidentally deleted all those files. So, and then because Google has revision history, we're like, hey, friend, can you upload it back up? So clearly, we're not very good at how to do this. But in thinking about the future, we have 300 labs, but in addition to the 300 labs that are building Miniscopes, there are many labs that are on the computational end. So one really great thing that has happened from the data being uploaded onto the website is that there have at least been 3 labs that have used the same data set and developed different analysis scripts, and then they send us kind of the results, and we've been able to compare and really compare apples to apples, and that's been very cool. We should probably publish that at some point in time. So the other thing that we're putting a lot of effort into is developing a software. So right now our acquisition software is written on C++. Our analysis software is in MATLAB, transitioning to Python. It's kind of all over the place. So in our new developments with the Miniscope, we're actually trying to move everything over to Python. The software will be for both data acquisition and data analysis. That allows at least the labs that are using Miniscope to index the data in the same way, log data in the same way, and that also allows us to do some online processing during data acquisition, so we can do some closed loop feedback experiments. So this is kind of the vision of where we're hoping to go, and hopefully with the help of the Allen.
Great. Thank you very much, Denise.
Thanks.